Nice. I really appreciate you all being here today. And I'm excited to sort of share some new research or sort of a new direction that our research team is taking in Ecuador. So with that, I will start sharing my screen. Can everybody see that okay? Okay, I saw Lauren's head nod. And are you seeing all of my slides or just my presentation slides?
We see the sidebar. Now we see just the presentation.
Okay, great.
That's good.
Okay. So I'll be talking to you today about what I've been working on since the last time I spoke to you, which was, or in this forum at least, in 2019, I think it was April. And I'll primarily be focusing on the role of the environment in the spread of antimicrobial resistance. And so we'll save questions until the end, but at any point, if you need me to clarify something, I think you can just put something in the chat, and then maybe Jen or Lauren can interrupt me and I'll try to clarify something. So, you know, historically, we've generally observed the introduction of a new antimicrobial, and, you know, following the introduction of a new antimicrobial, we often see resistance to that antimicrobial. So you see the year that a drug was introduced sort of on the top of the timeline, and then you also see where resistance was first observed. And so if we take the example of Staph aureus, you know, we started using staph in the 40s, and then 15 years later, we saw, we started using penicillin to treat Staph aureus infections in the 40s. And then 15 years later, we saw penicillin-resistant Staph aureus begin to crop up. Then we started using methicillin in 1959, and it was in the late 50s, and it was effective for penicillin-resistant Staph aureus. But then, you know, about 13 years later or so, we began to see methicillin-resistant Staph aureus, and that appeared in many countries. And then we started using vancomycin. And so and that was like in the early 70s. And then we saw vancomycin resistance occur, you know, in the 80s. So this is just a common phenomenon, and we're kind of in a race to develop new antibiotics and also preserve the ones that we have available now. So there's a lot of different types of resistance. And I just wanted to kind of let you know that there are a lot of really important areas in the study of antimicrobial resistance. You know, we have TB, we have Neisseria gonorrhea, we have methicillin-resistant Staph aureus that I mentioned, vancomycin-resistant enterococci, C. difficile, or what people call C. diff. Even we have insecticide resistance in insect vectors. So, but what I primarily focus in on is this order of bacteria called Enterobacterales. So it's an order of bacteria that includes Salmonella, includes Klebsiella and E. coli, that commonly cause infections in healthcare settings. So bloodstream infections, UTIs, and diarrhea. Now, there's indiscriminate antimicrobial use in humans, so inappropriate use of antibiotics in humans, and there's also a lot of inappropriate antibiotic use in animals, mainly for improving growth promotion and also for helping animals convert feed to weight more efficiently. And so that use, coupled with this increased global connectivity, has really facilitated the transmission of these drug-resistant Enterobacterales that I'll be talking about. So in the late 90s, we began to see more resistance among these infections to third-generation cephalosporins. So this third-generation cephalosporins, it's a broad-spectrum antibiotic. And these type of bacterial infections are often resistant to multiple drugs. So not just a third-generation cephalosporin, but they're also resistant to things like fluoroquinolones or what you might know as ciprofloxacin. So, and you know, these organisms, these specific organisms of ESBL producing E. coli or Enterobacterales, I should just stick with the order, we've seen a global increase in these in terms of prevalence, and there's been an 8-fold increase in global carriage of these organisms in the last 2 decades. And so there's a lot of concern about the increased colonization of humans with these all over the globe. And what happens when we have resistance to these 3rd-generation cephalosporins is we have to begin relying more on last-line antibiotics. And carbapenem is a type of antibiotic that's considered a last-line drug. So it's when we have resistance to 3rd-generation cephalosporins, or also called these ESBL producers, we then begin, if we have resistance to those, then we begin to rely more on these last-line antibiotics. And so we begin to see resistance to carbapenems starting in the early 2000s. So this is something that's popping up more in the news, these multi-drug resistant infections. So, and colonization with these organisms, they're not all going to cause disease. So a lot of these are commensals. So a lot of people are just colonized with ESBL producing Enterobacterales, but there have been studies to show that even being colonized puts you at an increased risk of mortality. And so it's estimated that there are about a little over 9,000 deaths in the US due to these specific organisms, ESBL producing Enterobacterales in the United States. And then this review, this systematic review paper basically looked at a bunch, 17 studies and showed that just being colonized with these organisms put people at a greater risk of mortality compared to those that were not colonized with those organisms. So even though these are not necessarily always pathogenic, we do see that colonization with these organisms is important. So this is, let me just say, I was just telling Jen and Lauren that this is my first, we're kind of changing directions with our research. So I've just kind of finished this presentation about 5 minutes ago. So I'm sorry if I'm kind of jumbling some of my points here, but hopefully just let me know if something's not clear. So in addition to human use, as I mentioned, we use a lot of antibiotics to raise animals in agriculture. And we also have, these active antibiotic compounds that are released into the environment from both human and animal use that ends up in the wastewater that can select for drug-resistant bacteria. And so what you're seeing here in this set of panels is basically that treatment with antibiotics leads to pervasive changes in the composition of the human and non-human microbiota, and it's encoded resistome. So the resistome is kind of all the antimicrobial resistance genes present in one's microbiota. And so this schematic depiction presents the changes in species diversity and antibiotic resistance levels pre-treatment. So you have pre-treatment on the left. So you have high diversity, low resistance, followed by the middle panel where you have antibiotic treatment and you see a significant reduction in the diversity of organisms in the gut. And you see resistance jump up in terms of resistance genes. And then you also have a partial recovery in that third panel on the right, where the microbial community structure and the resistance levels upon, after the cessation of the treatment sort of goes back to this sort of middle area. And I'll show you some more data that kind of demonstrates how this can kind of work. Now, there have been in vivo studies that have demonstrated the transfer of antibiotic resistance genes within microbiota, even in the absence of exposure to antibiotics. So it's not always that antibiotics are present. We see these resistance genes moving around between bacteria. And this drawing highlights how plasmids, so plasmids are these these round pieces of their extra chromosomal DNA, so not part of the chromosome, but they're self-replicating. And so how plasmids, represented by the round circles at the bottom left, can transfer resistance genes between different genera of bacteria and different species. And there are epidemic plasmids that are efficient in both collecting and also transmitting these resistance genes across bacterial communities. And these resistance plasmids have been found to move quickly all over the globe. And there, as I mentioned, there are these epidemic resistance plasmids that we're really worried about, and these have the ability to acquire resistance genes and then rapidly disseminate among species in the same family of, in this case, Enterobacteriaceae. So in this study, researchers, this was in Minnesota, but researchers longitudinally studied Enterobacter. It was Enterobacter cloacae, carrying these plasmids that were harboring resistance genes that encode resistance to last line antibiotics. So some of these genes code resistance to carbapenems. And so they did this really, thorough, intense surveillance, and they did whole genome sequencing, and they were able to show over time that these that the non-carbapenemase producing E. coli ST131 isolate, so at the bottom, which is ST131's a really bad organism in terms of its pathogenicity and ability to cause disease in humans, it had acquired a co-integrate version of the plasmids of the Enterobacter that we saw in the upper left-hand corner. So we see, this is the sort of scenario that we really want to avoid, where you might have these plasmids carrying lots of resistance genes, and they may be in a commensal organism that they can be transferred to something that does cause disease, and then we can't treat that disease because now it's multi-drug resistant. So now let's talk about a little bit more about how the environment is driving this. So this is a study, I like this study. It was, it's one that was conducted at UCSF among travelers, I think flying out of SFO. And the researchers studied a small set of individuals who traveled internationally. And they did a pre-travel fecal sample, a post-travel fecal sample, and then they followed them for 6 months, getting samples over the 6-month period. So they did it 30 days and then 6 months. So I'll try to walk you through these three panels. So in the panel on the left, basically the researchers showed that gut diversity stayed about the same post-travel, and then 30 days and 6 months after travel. So no significant difference, basically, in terms of the diversity of the organisms that were present in the gut or present in the feces. And then in the middle panel, they noticed that E. coli, that species of organism in the gut, increased across all travelers. And so it was statistically significantly different. And so the travelers are on the x-axis. And so if you go to the x-axis, you see traveler 7, 8, traveler 5, traveler 10. And so if you go to the top, you can see E. coli, and that color represents its increase. And then in the right panel, the researchers showed how the number of resistance genes, so this is kind of all those resistance genes that encode resistance to these drugs that are present in fecal samples, increased post-travel. So these travelers come back, they're carrying more antimicrobial resistance genes in their fecal microbiota, and then it drops back down after travel with an exception. So it appears that the ESBL and the AMP-C resistance genes remain high. And those are the ones that I've been sort of talking to you about. These are very important clinically. And so this suggests that even in the absence of selective pressure, there's not necessarily a disadvantage to bacteria holding on to these specific resistance genes. The other key point about this slide is that the environment matters. So, what is it though? These travelers, they traveled to different countries. Most of these countries were low or middle income countries where water, sanitation, and hygiene infrastructure is weaker. And so, but what is it actually that's driving this when, among these travelers? And that's the question that our team is really working to try to solve. want to tell you about one other study that just recently came out, and I thought it was kind of interesting. This is research that was done in the Netherlands, and so they're basically looking again at a few dozen travelers to understand colonization of travelers with these ESBL producing Enterobacterales on a daily basis. So they had these travelers, they were traveling to Southeast Asia, and they got fecal samples every day that the traveler was there, and to look at what organisms they're carrying on a daily basis. And so for every traveler, they got multiple isolates from the fecal sample. And in some cases, the traveler carried the same type of resistant strain of Enterobacterales many days during their travel. But in other cases, such as this traveler that I've highlighted with the red box, traveler number 8, there were colonization events where travelers picked up new strains of these ESBL producing Enterobacterales. And so it was very dynamic phenomenon, and they picked up new drug-resistant bacteria. And you can see that different days had they had different sort of strains carrying different types of resistance genes. So it's not necessarily you travel, you pick up this one organism, but it's a very dynamic sort of set of events that's happening. But the question again is, well, what's going on when these people are traveling? Are they recreating in contaminated water? Are they eating unwashed produce? Are they eating potentially contaminated meat and poultry? So that's, again, what we're focused on in our research. So this is, you've now seen about a dozen slides or so of background. Maybe that's too much, but I'm now going to begin talking to you about our research in Ecuador. And I want to acknowledge that I work with awesome people who I've learned a lot from, and I'll be sharing some research from findings from our collaborative effort. And many of these, I've had many awesome Berkeley students involved in this work, which was slightly affected by the pandemic. So I first started working in Ecuador in 2011 after I received an NIH K01 award. And it was through Fogarty. And we initially were focused on the spread of enteric pathogens between domestic animals and children. And enteric pathogens are those organisms that cause diarrhea in humans. And there are more than 1 billion people who raise domestic animals across the globe. And our team felt that this was really an understudied exposure in terms of domestic animal production. So you can see here just a backyard production of pigs in this case. So in this study, these earlier studies that were part of the K01, we identified relationships between the enteric pathogens in domestic animals and then those same enteric pathogens that we found in children. And we found strong genetic relationships. And in this case, on the right, what you're looking at is Campylobacter jejuni that we found both in children, and then we also found in domestic animals. And we basically, what we did for this analysis, what you're looking at is we analyzed 7 conserved genes in Campylobacter jejuni and looked at the sequence of those nucleotides in each of those genes to look at how related these strains were. And so you can see the overlap between isolates from children and isolates from chickens, also guinea pigs, and then I think rabbit, and then a dog as well. So we saw lots of overlap in these enteric pathogens. And then we also use these similar methods to identify relationships between pathogenic strains of E. coli. And while you might be thinking, well, no duh, like, of course, there's going to be exchange of these organisms between humans and animals, especially if the animals are defecating in and around the house. There were still ideas present in the literature that suggest that these organisms evolve in a specific habitat, in a specific species of animal, and that they can't easily be transferred to humans. And so we wanted to use some of these molecular tools to better understand kind of that exchange. Are there any questions or clarifications I need to make at this point?
There's two questions. One about colonization of ESBL, increased risk of mortality from COVID-19. Has that been studied? I don't know if you want to talk about that later. And then what is the rationale for colonization increasing mortality?
Yeah, I mean, I might tackle those at the end because those are pretty big questions, but there's a lot of mixed, I'll say first that there's a lot of mixed findings in terms of antibiotic resistance associated with the pandemic. There's a study that just came out of France that showed they saw decreases in infections caused by ESBL producing Enterobacterales there, but it's mixed because in some places we're seeing overuse of antibiotics associated with these, respiratory symptoms. Yeah, and I think I'll tackle that other question. That's a really important one after this.
Great.
Okay. So while we were doing this molecular microbiology research, we were noticing that there was a lot of veterinary shops that catered to small-scale food animal producers, and that many of these shops sold antibiotics over the counter. And there's a high concentration of veterinary pharmacy shops within our project site. And it's roughly, it's like roughly maybe 5 square miles where our study site is located. And we identified over 70 veterinary shops that sell antimicrobials over the counter. And that was actually work done by Zach Butson-Dozier, who's a PhD student in Epi. And we also began to conduct qualitative research, and that was also done by a doctoral student, who's a doctoral student now, was an MPH student, Chris Lowenstein. And basically that qualitative research suggested that many households, when we first were getting into this issue of antibiotic use, were relying on antibiotics for this backyard food animal production. And so based on this work and some preliminary findings of antibiotic resistance spread between animals and children, we were able to get an R01 to look more closely at the spread of drug-resistant bacteria between domestic animals and humans. So just in case you're wondering, I don't know if you can see that, but that is not a cigarette. People often see that picture, it's just a lollipop. So in case you're wondering that. Anyway, so in this research, in this R01, we continue to focus on this relationship of the bacteria in children and domestic animals. And we identified clonal relationships between ESBL producing E. coli shared between humans and domestic animals using a pairwise SNP analysis of core genome sequences. So this is using whole genome sequence data to show these relationships. And so we identified the presence of isolates with clonal relationships, which we call C. So you'll see in the figure, it says CR. And so every time you see a letter there, that represents a clonal relationship where it's basically nearly an identical ESBL producing E. coli. In this case, almost over 90% of the ESBL producing E. coli in our population, it's caused by these BLA-CTXN genes. So it's not a lot of diverse resistance genes that are driving this type of resistance in our community. And so the presence of isolates with clonal relationships and these BLA-CTX genes in children and domestic animals, again indicated that there's a clear connection between the drug-resistant E. coli from these different host species. And one thing that I want to highlight here that I thought was surprising, and some of you who study infectious diseases may not find it that surprising, but was this finding of finding clonal relationships that existed over long distances, so kilometers, suggesting that there may be a shared source of ESBL producing E. coli in our study communities. We assumed that we would see transmission mainly at the household level. And you can see that we did find that. So for the clonal relationship L that you can see in the top right of this figure, you can see that a child and chicken in the same household shared this exact same ESBL producing organism. But in many cases, these organisms were being shared over long distances in the community. And the ESBL genes, so these genes that encode resistance to third generation cephalosporin, so these genes that are driving ESBL production among the E. coli from both children and domestic animals, were mainly driven by the same allelic variant. So now we're getting really detailed about the specific genes driving this, and it was BLA, CTXM 65 and 55. And the benefit of finding these two allelic variants is that they can help us now home in on the source of these organisms and understand how certain genes and plasmids become dominant in the community. Now, in the summer of 2019, we had two MPH students who were part of the IDB division go down to Ecuador and work in the lab and also do some field work. That was Katie Kurowski and Rachel Marusinek. And Katie conducted a study of social and environmental determinants of children being colonized with these ESBL producing E. coli. And she did not find any strong relationships between our risk factors of interest and a child being colonized with ESBL producing E. coli. Still a very interesting study, and we found that both children and animals, the prevalence of these organisms was about 12% in both animals and children. And then Rachel did a different study where she assessed how the knowledge, attitudes, and practices of the primary caregiver to a child, so typically that was the mother, affected whether the child was colonized with these ESBL producing E. coli. And so she found very low knowledge about antibiotics in general and identified practices that would result in antibiotic resistance being a problem. So this is an area where we could intervene in terms of antibiotic use. But again, we identified very few associations between a caretaker's knowledge, attitudes, and practices and whether a child was colonized. And then we were also conducting some qualitative research to learn more about small-scale producers' knowledge, attitudes, and practices with regards to antibiotics and antibiotic resistance. And about half of the small-scale producers considered antibiotics important for growth promotion, and several respondents thought antibiotics were good for the health of their animals. But really, Zach, who again is in the PhD he's doing his PhD in epidemiology, sorry. He did a really interesting study using a simulated client methodology where we sent actors, these were veterinary students that were studying to be veterinarians in Ecuador. So we gave them a script. And we do this oftentimes in other things, like to see if, shops will sell alcohol to teenagers, where we send in these simulated clients. And so he basically went into we sent in these veterinary students with these scripts, and we wanted to look at whether these shop owners were promoting and advocating the use of antibiotics. And when you walk into these shops, this is kind of what you see. You can see antibiotics displayed right there in front of the counter. So you have ciprofloxacin and penicillin just right, basically 2 feet from your face when you walk in. And so what Zach really showed with his research was that there was really a lack of veterinary support or veterinarian support. So when animals are sick, there are few, people can't access veterinarians to really understand why their animals are sick and to get a proper prescription. We also saw that antibiotics were prescribed inappropriately. So among, the first initial recommendation that shop owners made when we gave them the script, and the script was basically went something like, I own 4 or 5 chickens and they're not growing very fast. You have any recommendations? And then we just leave it open-ended to see what they respond. And basically they do this whole scenario, then they leave, they go in their car, and then they fill out everything that they remember from that, simulated client visit. But, we also did, we did surveys in the households and found that 1 in 5 were reporting antibiotic use in animals in livestock and poultry. And that was over the last 6 months. So that kind of actually surprised us, that we realized that maybe it's not as common as we thought. And then we also saw very little use of these extended spectrum beta lactams, or these 3rd generation cephalosporins. So this began to sort of, erode our ideas that animals were the main source driving the transmission of ESBL producing E. coli in this community. So we now are finally to kind of the new direction that our team is headed in. And so we are again conducting our research out in sort of peri-urban communities of Quito, Ecuador. So if you know Ecuador, you fly into the capital city there, our research site is right by the airport. And so in our most recent study, we found that irrigation water was contaminated with ESBL producing E. coli, and that produce, primarily strawberries raised in our study site, were commonly contaminated with ESBL producing E. coli. And so the ESBL genes driving the ESBL production among the E. coli from the irrigation water and from the that we identified in the produce were again mainly driven by these same allelic variants, CTXM genes 65 and 55, that we commonly found in children and domestic animals. And we also, in survey data, we looked at, what water the backyard producers are using to water their animals, and they are using this irrigation water to use animals. And of course, animals can just go up to the irrigation channels and drink this water. So our team now is kind of moving toward testing the hypothesis that domestic animals who often drink irrigation water get colonized and amplify these CTXM genes. And we also are hypothesizing that despite having high quality drinking water in the household, so households have piped water under pressure that comes from glacial melt, and it's chlorinated. And so we haven't found a lot of fecal contamination in drinking water. But despite having high quality drinking water, we feel that community residents are exposed to these ESBL producing E. coli from both domestic animals and also contaminated produce. And so this was a paper that we just published that came out last month. And this is a big deal globally, as we kind of started really getting into this topic, but it's estimated that 10% of all irrigated land worldwide is irrigated with wastewater impacted water. And so this fecally contaminated water can be contaminated with wastewater that includes waste from hospitals, from household toilets, from meat processing plants, etc. And so this is commonly used to irrigate land. Now, 3 weeks ago, we submitted a proposal to NIH to look at the role of wastewater impacted irrigation for spreading ESBL producing E. coli in our community, with an added component of looking at human clinical infections in the same area. So trying to capture clinical infections that are caused by ESBL producing E. coli, to understand whether there's a link between what we're seeing in our community in terms of colonization and whether we're seeing a link to these clinical infections with ESBL producing E. coli. But I just wanted to show you that you see many of the same CTXM genes. These are the genes that encode for resistance to 3rd generation cephalosporins that are present in E. coli of children, domestic animals, and also irrigation water and produce. And this is a field of strawberries here on your right that's in our study community. So we hope to continue looking at the genetic architecture around these resistance genes to better understand the plasmid types carrying these genes in E. coli isolated from our study area. And then our plan is further analyze the relationships of the plasmids and other genetic elements. Like you can see, some of these are insertion sequences involved in the mobilization and dissemination and expression of these resistance genes. So here's kind of where we hope to prospectively sample water from high quality irrigation channels. So the circles in black are kind of the high quality irrigation waters that are closest to kind of the Cordillera, or sort of the mountain range coming down from the mountains. And so the irrigation water there is fairly clean. So we'll confirm that by testing for E. coli in that water, and then also testing in irrigation channels where after it's sort of run through many of these towns. And so, this research, again, I think will be relevant to many communities across the globe that lack wastewater treatment. And our hope is to continue working collaboratively with local stakeholders. We've been working with Agrocalidad, which is kind of the Ecuadorian version of USDA, to identify feasible solutions and point the way towards novel mitigation strategies that can reduce the health burden associated with the spread of antimicrobial resistance. And again, I think this is also relevant to the US, where we have several outbreaks each year that are associated with produce contaminated with fecal pathogens. And so these are some of the different species that fall within the order of Enterobacterales that I've talked to you about. In 2020, these are outbreaks that were associated with produce. And we've also seen studies from Europe that show that, in studies of vegetarians, that vegetarians are actually at an increased risk of carrying ESBL producing E. coli and Klebsiella, in contrast to non-vegetarians, which are the reference group in this analysis. And so we do think this is an important pathway of exposure to these multi-drug resistant organisms. And we want to, really identify specific ways that we can mitigate these exposures. And so with that, let's see how I did 12, 18. If we have time here, I'll take any questions or comments.
Great, thanks so much, Jay. That was really interesting. There was one question that came in: do humans who consume poultry treated with antibiotics develop infections that are resistant?
Well, we should actually, I hope Lee Riley is on, because this is an area of work that Lee Riley has been working on, and many, many studies on this topic. Lee, do you want to chime in on that?
Sure. We actually did a study a long time ago here in Berkeley campus. And this was done by Amy Mangas, one of my first PhD students. And we found that not necessarily drug-resistant E. coli that caused UTI, but just UTI itself was associated with consumption of chicken in the previous 2 weeks of the episode of UTI. And so, so I don't know if it has to do with necessarily with chicken given antibiotics, but there is an association with UTI with recent consumption of chicken. And the other association we found was frequent alcohol consumption among students. But now that I have you, Jay, can I ask a question, Jen?
Yeah, just so long as it's not too difficult.
No, it was a very nice presentation. Glad to see you getting into this new area, looking at produce, because as you point out, that's really important, even here in the US. But you did the whole genome sequence analysis, and a lot of the E. coli you identified that had ESBL genes. Did any of those E. coli strains belong to the known so-called extraintestinal pathogenic E. coli lineages?
There's a lot of diversity in sequence types we found. We did find some ST131, ST95, so some of the, and I think 69 as well. but the majority are not ones that are driving sort of this, this sort of scourge of antibiotic infection.
That's really interesting. Okay. And you found ST95. That's actually very interesting because here in Berkeley, ST95, as you know, is the most common ST that found in students with UTI, but it never has ESBL gene. So.
Oh, interesting. Well, let me look back at that. But I think in our irrigation water and produce samples, that's where we identified the ST.
Okay. No, that's really interesting. Okay, great.
Thanks. And Scarlett has a question. In that last study, you talked about why are vegetarians at higher risk of carrying ESBL, E. coli? Is it because the antibiotics are already restricted in meat production where that's, or weren't carrying AMR genes, or why?
That question didn't really dig into, they were basically looking what risk factors are associated with someone being colonized. So they didn't really get into specific exposures. But my bet is that there's so many other pathways of exposure that beyond meat and poultry, though while I think that's a very important exposure pathway, in this case, it could be that these vegetarians are, exposed to a lot more produce that's maybe produced organically, and maybe they use a lot more animal waste for as a fertilizer. And it's not necessarily, it's interesting because resistance is not always linked up to antimicrobial use. Sometimes it's because we have this leaky system where these organisms can spread, and even though there's no sort of selective pressure for them to be there, they have spread, and there's not really any pressure for them to lose these genes once they have them in many cases.
Thank you. Two more questions. Darlene has a question. What would sufficient control groups when we think, oh, Darlene, do you want to say your question? Go ahead.
Let me unmute. Well, it's a follow up to what other folks are talking about and what Jay was. It's nice to see everybody's faces. How do we? It's so interesting to see this data, and again, it leads to more questions about how do we think about this, right? And a comment following mine is about multiple pathways, which is always the answer, but sort of the more refined, the better we ask the questions, the more complicated the findings. And it leaves me thinking about how should we be thinking about this? And it's what you were just talking about in your response to the previous question. Like, how do we know? How should we be thinking about this? What should we take away from this? I'm a happy carnivore, so I'm happy.
Well, I do think it is kind of very complex and messy in terms of all the exposure pathways. I think, the general, if we could get back to sort of a general thing that we've understood over the ages, is sort of keep feces out of our mouths, at the most basic level, but then understanding all those ways that we can intervene, especially in places where there's not the resources necessarily for wastewater treatment plants. So coming up with alternative ways to intervene, and that might be interim solutions, but maybe get us, part of the way there, kind of like masks do for COVID-19. So, that's what we're really trying to figure out. How do you, at different levels, maybe of economic growth, what do countries do to intervene to stop this? There are also really cool methods coming out. And, for this grant, I didn't mention this, but, I've roped in Joe Lunar because we're trying to, there's some new methods where they're using bioinformatics and some really sophisticated analyses to understand sort of how these organisms evolve, and what they've done this in hospitals where they track the movement of these organisms in hospitals, even after they've, gone through some mutations. And so we're going to try to apply some of those methods that they've used in hospitals, but apply it to a community setting so we can really understand what is the source and where did it all start from, and really kind of home in on where we can intervene.
Thanks.
Good to see you, darling.
Let's see, Layla has a question. Are the drugs usually high quality? Could there be counterfeit drugs, and how might this affect things?
Yeah, I mean, Ecuador is an upper middle income country, and I do know that drug quality is an issue in global health. I know that there's a lot, but I don't necessarily think that it's an issue here. Maybe, it could be a risk. It could be a risk because, the drug quality is not, maybe it has less of that active ingredient. And so it might be creating more of a selective pressure and not, effectively treating the infection at the clinical dose that you need. But I'm not, it's a good question. It's probably something I need to, look more into in terms of understanding drug quality there. But I think, yeah, I don't know the answer.
Great, thanks. It's all right. And Karen has a question. Children and adults in Ecuador typically have a lot of contact with their animals outside the house. Did you also examine animals kept inside, guinea pigs, et cetera, who are often raised in the kitchen, and whether indoor household or kitchen contamination might also be a problem?
Yeah, it's interesting. Our communities don't allow pets into their home. And dogs are the primary pet that animals own. It's, over 70%, and they're used as a guard animal. And at night, so these dogs do roam freely, but we don't see a lot of contamination inside, or we don't see a lot of domestic animals inside the homes. And that's different than a lot of communities in other countries around the world.
Great. Let's see. I think one other question early on. I'm not sure the rationale for colonization increasing mortality. I don't know if you wanted to address.
Oh, yeah.
2 minutes and then yeah.
Well, I think, basically what happens is that even if you have these ESBL genes in an organism that's a commensal, so that it's not necessarily a pathogenic strain, just as Lee mentioned, like ST95, he said that, we don't see the ESBL gene in ST95, but there is the potential, the gut microbiota is a perfect place for these genes to be shared. And so when you have this resistance that is present, that means that you have to then move towards an antibiotic that's closer to a last line antibiotic. And after carbapenems, you end up moving to a very toxic antimicrobial or antibiotic called colistin, which is nephrotoxic, so toxic to your kidneys. And so, it's probably, I don't think they looked at the details of how people in all of these reviewed studies died, but it could be that they're, it could be they're just in a worse case, but many people do end up dying from ESBL Enterobacterales infections. And so the CDC estimates over 9,000 people die of those kind of infections that can't be treated.
Well, any other questions? And we have 1 more minute. Thank you so much, Jay, for sharing your work. And it's really interesting to hear about the new line that you're taking. So thank you very much. Appreciate it.
Yeah, thanks. Good to see you. Thanks for coming again.
Thanks everyone for joining. Have a good day.
Bye.
Bye.